Dr. Indira Turney, who is an associate research scientist at the Taub Institute for Research on Alzheimer's disease and the aging brain at the Department of Neurology at Columbia University Medical Center. She earned her PhD in Cognitive Neurosciences from Pennsylvania State University, and her current research explores multiple neuroimaging modalities to investigate the Lifecourse social-cultural and structural mechanisms that influence brain aging and racially and ethnically diverse adults, particularly in midlife and older ages. Her primary objective is to investigate how racism becomes embedded in the body and brain, particularly impacting overall health. Today, she's going to present her work on understanding of core dynamics of racial ethnic disparities of brain aging. Please join me in welcoming Dr. Indira Turney.
Thank you for that, Francisco. Thank you guys for coming here and having your lunch with me, or while I speak. I'm really excited to share some of my recent work with you guys, and feel free to ask questions in between. If you prefer to stay at the end, that's fine as well, but you're also welcome to ask during. All right, so first I'll kind of take you guys through my personal research journey, especially there's a lot of students in here, and I thought that it's always helpful for me to understand like where people came from to understand how they got to their work. Because I think when I see speakers up here, you guys are probably thinking I'm brilliant, and I am a little bit, but like it was, quite a journey, and I don't want you guys to feel like, oh my God, how did she get here? Because it's very much possible for each and every one of you. And then I'll talk about like my overarching research aims, recent findings from current studies, and then just like future plans of what I generally plan to do. So ultimately my goal is kind of to explore how these biological, social, cultural, and these like structural factors kind of intersect to contribute to brain aging and dementia across diverse groups. In this case, a lot of my work will be focusing on race and ethnicity groups. But again, like I want to take you guys through this. So I first started with my undergraduate degree at the University of the Virgin Islands. I'm originally from the Caribbean. There, I barely knew what research was. I was a McNair scholar, so that allowed me to do summer programs. So like I went to like University of Iowa and Ohio State to do like a 6 or 8 week, I don't remember, it seems much longer, but about 3 months of research at another university. And that's how I kind of like figured out the different things that I wanted to do. My first research project was actually trying to understand how people select their mates in college, while also accounting for alcohol consumption. Because I was trying to figure out, like, are people just picking partners based on, because coming from the Caribbean, we do drink a lot, or they drink a lot. And so I think it was just like one of those things like, oh, does this play a role in who you choose for your partner? And someone allowed me to do that. But I realized, okay, the questions was not really what I was interested in, but I really enjoyed like the statistical experiences, the methods that I used to answer the questions, creating a survey, asking people questions. And then that led me to like my next experience at Ohio State the next summer was more brain related, because I was like, okay, but I really want to understand like what's underlying decision making. And so then that turned into a project with looking at multiple sclerosis, older adults, and brain aging. So not exactly what I thought. And then I was okay, I don't really care about multiple sclerosis, but I do admire the aging process because I've always had an interest in that. So then that led me to like narrow down things more. So the story of that is like, try everything and you'll figure it out on the way. And then, so even though my undergrad didn't have research experiences, like this allowed me to like get experiences at on the mainland. And then I applied to grad school twice. The first time I did not get in. And then I ended up doing a pre-doctoral program at the University of Pittsburgh, where essentially was just the first year of grad school where I got to do research. I got to take courses. So I took some stats courses. And then also just like, again, narrowing down my interests. So here I did like brain aging and exercise. And then that led me to reapply to grad school. Now I have also a better network. It's really not about, what is the saying? It's not about what you know, it's who knows what you know. And so this time around, I think it was a lot different in the sense of like my mentors knew who I was. They were able to communicate that to other mentors. And so as a result, like my applications were a lot more specific and a lot more, aligned with the places that I were also applying to. And then, so I decided on Penn State based on the experiences of the research topic. And there, I had an amazing experience a lot of times. So here I changed into, so it was no longer exercise and aging. This was just now me focusing on aging in young and older adults. So here I kind of use this as like the foundational experience of understanding aging and using brain aging methods to do that. And then I wanted to now expand this to more diverse populations. And as a result, my postdoc at Columbia is now focusing more on like diversity and health equity in brain aging. And so ultimately, my goal, like I said, is to kind of, understand brain health equity and understanding these intersectionalities and how do these things get embedded in the body. And I also want to highlight here throughout, I have been supported by many NIH awards, but most importantly, DEI initiatives where every step of the process, there was some kind of initiative that exposed me to research that allowed me to like know what a PhD, like my sophomore year in college, I didn't know what a PhD was. And so I really understand the value of community and mentorship. And so I'm very passionate about that as well. And so I'm happy to talk about any of those initiatives after or whenever you feel inclined to. All right, so let's get to the science. So the goal of my work is really to, like I said, achieve health equity. And I kind of use this like public health framework, if you will, as a guide. So where if you think about health equity exists when all people have a fair and just opportunity to be healthy, right? Especially people who've experienced socioeconomic disadvantage, historic injustice, and other avoidable systemic inequalities that are often associated with like social positioning, such as race, gender, ethnicity, and so on. And that doesn't really currently exist. And so oftentimes when we think of these racial or ethnic disparities, we kind of think of the differences across ethnicity groups, but health disparities are kind of more so the preventable differences in the burden of the disease, the injury, the violence, or even opportunities for people to achieve optimal health. And so, and that is like specifically experienced by socially disadvantaged populations. And so in the context of health disparities, when I refer to this, I'm not just referring to differences in health, but rather to a particular type of difference in groups that have persistently experienced social disadvantage, discrimination, and so forth. And so it's really critical, I think, for us to really understand the different life course factors that influence and that are driving some of these inequalities. And so to really successfully tackle some of these things, we must really look at the structures and the systems that are really underlying the health behaviors and the health outcomes. I really do think like by addressing some of these upstream contributors, including distribution of power, reframing the questions that we even ask about health would be important, and really prioritizing health equity in the sense of meaning to like think about our work differently so that we can really create better conditions for health. So some questions I consider when I'm taking on this approach include, how do people age across different groups that are at risk, or do some people age faster than others? What are the risk and the resilience factors that are associated with such aging processes? And in some cases, what are the protective factors that do that? And importantly, ensuring that we're also studying the right people so that the data represents the people in our communities. And so this kind of leads me now to the overarching aims of my research, which is first to examine the disparities in brain aging and dementia among these racially and ethnically diverse adults, then to kind of determine the life course factors that impact brain aging within these diverse groups, which ultimately helps us better understand the causal pathways and the mechanisms that are underlying these disparities in order to really, advance health equity and inform these targeted interventions that would address some of these disparities. And this is important because there are well-documented disparities in dementia risk across racial and ethnic groups, as we can see here. So in this figure, you have age on the x-axis and incident of dementia rate on the y-axis. And we see that African Americans in the purple jagged line were 65% more likely to develop dementia compared to Asian Americans in the black solid line here. And so I really do believe that a critical first step in even eliminating some of these disparities is first to just understand, well, maybe not the first step, but overall is to like understand these mechanisms so that we can really intervene and accurately identify these disparities because the causes, the risk, and the resilience factors, and even the course of ADRD will vary by groups of people. And so we really need to like take a more precision approach, if you will, to understand aging and disease. So like really understanding as much as we can about someone's risk and their resilience, to really ensure that we are developing the right interventions at the right time for the right people. So I also want to highlight some of these gaps in the literature that my work aimed to fill. So a lack of diversity in study participants, and really up until recently, most studies have predominantly included white, highly educated participants, which kind of hinders the generalizability and really perpetuates this like biased results, providing pretty much an incomplete picture of brain aging and ADRD because we're such a diverse group of people. And that just further hinders our understanding of the unique characteristics and the disparities in different populations. Another gap here is limited understanding of life course factors. So research has also focused primarily on late life risk factors, overlooking early and midlife. And I know, especially with high school and beyond, like I know I'm speaking to the choir here a bit, but like overlooking these early to midlife experiences, which is crucial to understanding disparities. And this gap really limits our understanding of like how, when, and why these disparities in brain aging occur. And so, you know, trying to figure out when they emerge, because as we know, like life course factors play a really critical role on health outcomes. And then we also have biological and genetic factors have been dominating the research. As a neuroscientist, that probably sounds like I shouldn't say that, but I mean, it's the truth. But we do neglect the impact of social determinants, such as, you know, socioeconomic status, structural racism, which significantly contributes to these disparities. And so we have to account for those things in our research. And then limited intervention and research where, you know, effective interventions addressing disparities continue to, you know, be understudied, and highlighting this need for incorporating both biological and social cultural factors. And I really hope that my work, you know, is able to like inform these interventions by kind of incorporating a more holistic approach, by the questions I ask, the people I study, and the methods that I use to do that, to understand the overall process of aging. So my work focused on the differences and changes in brain structure and function, which kind of is measured as a proxy for neurodegenerative diseases. So using MRIs to kind of understand the risk factors, the progression of dementia across these diverse adults. So today I'll kind of present on a few of these biomarkers that I use in my work. And so here you see, for those of you who are a little less familiar, so in the first figure here, I'm showing cortical thickness, which you can think of as a measure of how much shrinkage occurs in the brain as you age. And then B and C here shows a measure of cerebrovascular disease, which are essentially tiny little strokes in the brain. So more of that is not good. And we refer to those as white matter hyperintensity volume. And then the last figure here shows resting state functional connectivity, which is more or less how multiple brain regions kind of hang together, communicate with each other, if you will, while the brain is at rest. And I use these markers really because they provide insights into the risk and the progression, like I said, for dementia and aging, but they also provide different measures. And we know that different measures can give us different information as to like what pathways and what processes that are actually happening for overall brain health. And we also know that that can vary by groups of people. So the more measures that we have that kind of capture different processes may be helpful. And I'll show some of that as we go along. The data that I'll present today come from two community-based aging cohorts in New York City, YCAP and the Offspring cohorts, specifically within the upper Manhattan, New York area. And really, you know, by spanning across multiple generations, these cohorts kind of give us more of a lifespan perspective, kind of facilitating a more comprehensive investigation into brain aging and dementia within these diverse groups of people. And so specifically, YCAP include participants who are about 65 and older, who are Medicare eligible participants in, like I said, the upper Manhattan area. And the Offspring cohort includes they're the adult children of the YCAP participants. So there's also the intergenerational effect there. And on average, they're about 55 years old, and YCAP on average is about 77. So about 20 years apart in age on average. So let's get into this. So in order to really understand, you know, the influence of these social cultural factors on brain aging, we kind of need to understand how this gets embodied. Like, how do these factors get under the skin, if you will, to eventually affect brain health? And so one way to explain this is using the weathering hypothesis, where this hypothesis proposes that the cumulative impact of like social, physical, and economic adversities experienced by especially Black Americans can lead to this early health deterioration, which is believed to kind of be more so caused by chronic reoccurring stressors. In other words, it's considered the wear and tear on the body. And this has been understood in other disease systems, but less so in dementia. For example, a study here by Geronimus and colleagues looked at allostatic load as a measure of biological aging, indicating accelerated wear and tear in the body. And this figure shows, so you have again here, age on the x-axis and probability of allostatic load on the y-axis. And results showed that Black men and women had higher probability of allostatic load, and Black women had the highest compared to all groups across age. And this also shows that the racial difference widened as they got, as they age, as age increased. We saw a larger difference, which can be for several reasons, and we can talk about that. But essentially, these results kind of show that how critical it is to really address these weathering in vulnerable populations. And lastly, before getting into the data, I like to show this figure of the progression of former President Barack Obama, because I feel like it highlights the potential effects of stress on aging, measured through the gradual graying across, his tenure. Although I still struggle with always showing this figure, because it's not the best way to portray accelerated aging, because personally, I think he got better with the gray hair. But then it also reminds me of this, another issue where individuals from minoritized populations, particular Black individuals, may appear to be aging normally, or in some cases, maybe even better. I mean, you probably heard Black don't crack, because it really doesn't. However, internally, we may experience accelerated aging. And so, I think that's another misconception. You hear oh, the Black participants are a little more hardier in our study. And, I don't think that's necessarily a good thing. It's outwardly, we may appear like we're aging normally, but we do show biologically accelerated aging. But in any case, this is an example of an individual who had all the resources that came with being a president of the United States. So now imagine what it's like for you and I without those resources. And so considering the intersectionality of SES and race kind of really underscores the importance of examining these factors to understand, what's really driving these disparities and overall health. And so to kind of tackle the first part of my aims, I'll first show kind of where I've identified some of these disparities in my work. So through my K9 R00 grant, I tested the weathering hypothesis to kind of identify disparities in brain aging among these racially and ethnically diverse midlife and late life adults. And what I found was that, so first showing race and ethnicity on the x-axis and cortical thickness on the y-axis. And so remember, cortical thickness is just shrinkage in the brain. And the first figure represents the older participants, so the Y-cap participants, and the midlife adults are the offspring participants, so the younger folks. And what I found was that racial and ethnic disparities in cortical thickness only occurred in late life. So we only see these differences in Y-cap and not in midlife. Where you see that the Y-cap participants, white older adults, had greater cortical thickness than Black and Latinx participants. And then in midlife, we see that the cortical thickness was more similar across race and ethnicity groups, kind of suggesting differential aging patterns across the lifespan. And then when I looked at markers of cerebrovascular disease, so again, the small strokes in the brain, a higher number is worse. I found that racial and ethnic disparities in white matter volume was evident across both midlife and late life. So specifically, we see that older Black adults in the Y-cap, we see that older Black adults had highest level of white matter hyperintensities, followed by Latinx participants, then the white participants. And surprisingly, in the midlife cohort, we see the same pattern, but the disparities were even more pronounced in midlife, which was very surprising to me anyways, kind of indicating this early onset of disparities in cerebrovascular health. And overall, these disparities were observed between Black and white participants were much larger than, say, the Latinx and white participants. And we can also dissect that if you want, for both measures, while the Black and the Latinx disparities were more similar. So they were more similar to each other. statistically anyways. So taking this a step further, when we looked at the association of age on these brain measures, so brain aging was greater in later life than in midlife for Latinx and for white participants, but not the Black participants. So you can see here showing again age on the x-axis, cortical thickness on the y-axis, the blue line representing the midlife younger folks and the orange line representing the later life older adults. We see that among Latinx and white participants, there's this inflection in the age slopes, right? So you can see here that the midlife brain aging suggesting that midlife brain aging is better than later life among these two groups. However, when we look at the Black participants, there was no difference or inflection in the age slopes between midlife and late life, suggesting this accelerated brain aging in midlife, middle-aged Black participants. The same pattern was observed for white matter hyperintensities at even, again, a greater magnitude where we see differential aging between midlife and late life for Latinx and white participants, but more similarity across slopes between the midlife and later participants who were in the Black group. So all in all, these findings kind of suggest that the disparities in brain aging may manifest even earlier than previously recognized. And I really believe that this is influenced by the social, cultural, environmental factors and the experiences that are unique to these minoritized population. And so the next step would be to kind of understand the underlying factors that are driving these disparities in aging. And so I turn to genetics to understand, these mechanisms that are driving these disparities. Because, after age, genetics some would believe that genetics would be one of the main driving factors that predict your health. And so focusing on ApoE4, the ApoE genotypes are strongly associated with dementia, if you're not familiar, with having two E4 alleles significantly increasing your risk by almost 50%. However, this map here I really like because it shows a wide variation in the geographical distribution of ApoE4 alleles with a higher prevalence among individuals of African ancestry. So looking at the bright red regions. Still, here we see that despite this higher prevalence of E4 alleles in African Americans, based on these graphs here, the odds ratio, so the risk for developing dementia, is much lower among African Americans compared to Caucasians and Japanese participants with the E4 allele. And so again, I took this to kind of suggest that, well, clearly there are other factors rather than genetics alone that may contribute to the disparities that we observe across racial and ethnic groups. And some of my work support this hypothesis, but just in case, I still wanted to, get the data, right? So I looked at the association between ApoE4 and resting states. So remember, looking at how the brain regions kind of hang together and how healthy that is, there's a certain expected pattern. And so if it deviates from that, you can tell whether or not that's healthy or not. So specifically focusing on temporal regions, so within the temporal pole, within this network of regions that are represented as called the default mode network, which is just how the brain generally is when you're mind wandering. And so in over about 500 participants, diverse racially and ethnically, in the older cohorts, so in YCAP, who are 65 and older, we did find differential pattern of ApoE on brain connectivity. So specifically, we saw that you can see that ApoE4, so the darker bars compared to the lighter bars, ApoE4 positive participants exhibited less connectivity compared to their ApoE4 negative participants who were white, but we found an opposite pattern in Black participants. So we saw a reduction in connectivity in the white participants, but an increase in connectivity in Black participants. And there's a whole other literature in that we can also talk about. But again, kind of just suggesting that there might be other factors contributing to the deficits that we see in these populations compared to white participants. So now to kind of focus on the second part of my aim. So my current and kind of ongoing future goals are really to kind of determine, again, what are the various factors across different time points in life that really contribute to some of these later life disparities. And so in addition to racial and ethnic disparities in dementia, as I showed earlier, there's also evidence for socioeconomic positioning disparities in dementia. So here, again, age is on the x-axis and survival of dementia on the y-axis by socioeconomic position trajectory groups. And what we see here is that socioeconomic positioning trajectory significantly impacted the survival to dementia, with higher SES associated with better dementia outcomes in the opaque circles. And these findings really suggest that differences in the burden of the risk factors, especially by SES, may contribute to some of the higher rates that we observe in these minoritized population, and the accelerated aging that we have observed in these groups. And so again, hopefully strategies aimed at reducing these disparities may favorably affect accelerated brain aging and dementia incident as a result. And so briefly, again, so the literature shows that, Lifecourse SES, so from childhood versus adulthood, current SES is an important predictor of health status, so like cognition, dementia, and may really help us understand what are some of these underlying mechanisms that contribute to some of these disparities. And so given that SES across the lifespan predict these health outcomes, I'm also curious about whether there's a period of susceptibility. And I know depending on your background, you may interpret that a little differently, but for the sake of bear with me. I also am interested in like, where is SES most impactful? Like, does your SES in childhood is most impactful versus like your current SES or SES in later life? Like, where does this, vary and potentially why, to really, again, help us get at these different mechanisms. And so to take a stab at this, I'm currently looking at Lifecourse SES measures on these brain aging in this midlife offspring cohort. So now looking at the individuals who are 30 to 55, and specifically looking at the role of childhood versus midlife SES on these measures of like small vessel cerebrovascular disease and cortical atrophy. So again, small strokes in the brain or shrinkage over time. And this time looking at it within each racial group. And so I use these direct measures of childhood SES, where I use a composite of the YCAP participants. So remember, they're the children of YCAP participants. So I use the YCAP participants' income, education, and occupation. And then this composite would be used as just an overall childhood SES. And then their current midlife SES would be their current income. Does that make sense? Okay. And so the goal here is to kind of really assess whether childhood and midlife SES differentially predicts brain health in these midlife participants. And so I'll present some preliminary findings today. So the blue represents the Black participants, green for Latinx, and the red for white participants. And the results are really showing that greater childhood SES is more strongly associated with lower white matter hyperintensity volume. But in this case, it was only in the white participants. And I also wanted to highlight here that, so the circles represent the proportion of each group at high or low childhood SES. So a larger circle represents a larger proportion within an SES group, and the slope or the steepness of the lines represent the strength of the effect. So in this case, the strength of childhood SES on white matter hyperintensity volume. So you can kind of see here, for example, that even if there's a small effect observed in Black participants, it's a higher number of people with higher white matter hyperintensities. And so if the goal here is really to reduce disparities, with fewer white participants in the lower SES group who are having a bigger benefit, to get a bigger bang for your buck would be to intervene on the minoritized group that show greater white matter hyperintensities. And so this is where sometimes I really think like, if we just rely on the stats, we like don't actually address the issues. And then in midlife SES, we see a similar pattern, but this time it's the Black participants who showed greater midlife SES to be associated with lower white matter hyperintensity volumes. And so I'm still speculating some of these findings and kind of digging deeper into the analyses, but I'm kind of contemplating whether these patterns kind of suggest some cumulative risk versus resilience, and kind of thinking more about how these structural factors may amplify the importance of midlife SES as a potential protective factor in Black participants. Again, I'm happy to discuss this with anyone interested. But altogether today, really my work, so I revealed a few key findings. So looking at differential patterns of brain aging across different groups with accelerated aging observed in midlife Black participants. ApoE4 risk does not fully explain the disparities, indicating other factors may be at play. And I also explored potential evidence towards critical time periods where structural factors like SES may differentially affect brain aging in midlife across groups. But my long-term vision is really, it's kind of anchored in understanding and addressing some of these cognitive and brain health disparities across diverse populations at risk, building upon, a theoretical framework established by ongoing efforts. And so this kind of leads me to quite the task, but I'll have a job for quite some time, hopefully. But I'm really excited to continue this work where I'll, dive deeper into some of the Lifecourse structural risk factors and the resilience factors like quality of healthcare access, robust social network, community, neighborhood quality, and kind of seeing if that helps us understand why some of these groups are excelling versus others, but also, again, some of these factors are protective, and really identifying whether some of these factors vary, which ones will vary by racial and ethnic groups, which is why it's really important to not just look at the intersection of race and these factors, because some of them may be different for different groups at different age time points. And then also identifying whether or not some of these factors are categorized within specific type of environmental factors versus cultural factors and so on. While also integrating some of this fluid biomarker work, because that's my background, with like advanced imaging methods to really kind of get at these identifying maybe earlier diagnostic measures to reduce disparities. And then lastly, also, oh, well, no, not lastly. So also like integrating more of a differential biomarkers through like understanding the biology, so including like DNA methylation and really getting at some of the internal stuff, but also like understanding how they interact with the external stuff, if that makes sense. And then lastly, but certainly not least, it's like I'm also very committed to deepening my engagement with like the community by really collaborating closely with diverse communities to understand like what are the priorities of the research so that they can inform us in how we're going about designing these studies and implementing these interventions and what's actually feasible in the community. And this way the goal would be to tailor these interventions and treatments with more precision by understanding the unique contributions of each group at varying time points. And I want to leave you guys with kind of just this like overall framework that I kind of try to like map my work onto with like 5 general steps to get through this process. So first, we kind of identify and examine where these inequalities exist. And I've kind of established some of that in my work. So for example, when I mentioned showing accelerated brain aging in midlife Black adults, then once these inequalities are identified, it's critical that we identify the factors that exacerbate as well as attenuate these inequalities. So at this stage, it's kind of critical that we include diverse participants across race, ethnicity, sex, SES, and so forth. And this is where I feel like a lot of the meat of the work is right now. And so here, like I said, I looked at ApoE4 and resting states, so looking at some of the genetic factors, kind of leading me to begin to investigate the structural and social cultural factors across the lifespan and not just in later life. And then around here, we see the third step is to examine the causal pathways and mechanisms, seeing how these factors interact to influence health inequalities, which, like I said, may be different across groups of people and within groups as well. Because again, the goal is really to reduce disparities with disproportionately affecting the groups of people that are at risk and disadvantage. Then we really must, figure out the direct as well as the indirect factors that lead to poor brain health in these groups. And then step 4 here is where our work acknowledged kind of the multiple forms of racism across various contexts as a main contributor of these inequalities. And at this step, we must really center the marginalized group experience, as they are the ones most affected and understudied. And so I really want to highlight here the importance of like, again, comparing not only across groups, but also within groups, and really understanding the unique experiences of these individuals to have less biased results. Which leads me then to step 5, where my work will kind of hopefully inform research on developing these biomarkers that detect these risks much earlier in the course of aging and disease. Because as you can see, we've seen some accelerated aging, but like especially if you're going to go with more minoritized groups, it's really hard to get people into an imaging scanner. And I don't think again, as a neuroscientist, that probably goes against everything that I've been taught, but I don't think we need to understand, to bring people into the scanner. Also, by the time we start seeing changes structurally, these things have already been happening for 15, 20 years, if not even much more. So I do think we do need to develop some earlier biomarkers, and it would be really nice if you can just go to a doctor's office and get a blood test and be like, oh, this predicts that you are at risk. And then here's these interventions that may or may not help. But really, you know, I think the goal here is then to like use that to also inform interventions that are aimed at reducing these disparities. And more importantly, going back to step one. So we identify the disparities, we figured out what's making them worse or better. We've looked at the different pathways, interaction with different environmental and biological factors, as well as centering the people who are at risk. And then hopefully that informs what kinds of interventions we should create. But most importantly is then test whether these interventions are actually reducing the disparities that we think that they're supposed to reduce. Because I think a lot of times we just stop here and we're like, oh, this is what it should do. And this is still another process. It's a very reiterative process. And that's kind of the goal I hope to get my work at. Again, kind of like ensuring that we're developing the right interventions for the right people at the right time. And thank you to all my postdoc lab mentors, because that's where I've developed most of this work, as well as the participants, of course, from Offspring and Whitecap, and my community network, as well as, of course, this is impossible without the right funding. And so I look forward to our discussion.
I'd like to reserve the first two questions for our guests. Thank you so much for this presentation.
Thank you.
You had mentioned, I think, that with Black women were at the highest risk of being dementia-related diseases. I was just wondering if you had noticed like any gender differences in how any of the Lifecourse factors might be manifesting disparities.
No, that's a really great question. So this is current stuff. So it's so much to explore when you're looking at the intersectionality of like race and sex. And so even just thinking of SES, like what does current income mean for a man versus a woman? It's very different. What are your responsibilities? And so this is where, again, I'm really interested in looking at how do these things, what access does it provide to you? Because making, say, $50,000 as a man represents something very different than a woman making $50,000 in a specific city, given other responsibilities. But also the types of professions, this kind of like, what does that do on your day-to-day body reactions as a man making that amount versus a woman making that amount? So I don't know the answer to that yet, but I'm pretty confident that there are gender differences when we're going to look at some of these environmental and social cultural factors, because they just play very different roles in our lives and expose us to very different experiences. Yeah, it's a really great question. You're also welcome to ask me experiences about my journey if you don't want to. It's a grad student.
Can you give me a box off
For
oh, yeah,
this one. Mm-hmm.
So I'm looking at your brain regions too, and I'm thinking maybe like 3 vulnerable regions in here, right?
Mm-hmm. Yeah.
Do you think that maybe that's why I didn't see some differences in the offspring? Like, I guess I'm wondering, you looked at 80 vulnerable regions,
Mm-hmm.
took a more holistic approach and thought about neural reserve, not just in those 80
Mm-hmm.
vulnerable regions. Would you see a different pattern of that?
So I thought so, except so like here, when you're just looking at overall mean of cortical thickness, there's no difference by group within offspring participants. So I do think if that was the goal, yes, I would look at whole brain. But then when I showed this figure here, where you're still seeing differences in cortical thickness and white matter hyperintensity. So the younger people are still healthier critically than the older people in Latinx and white, but not in Black. But the mean level of like cortical thickness in Black, Latinx, and white are the same, but the trajectory is different. So like, you're right, like in later life, these things may deteriorate faster, but even still with something that should start deteriorating later in the Black participants, we're still seeing the midlife one having lower cortical thickness compared to the the older participants. But I do think like if I looked at whole brain, I would probably see some mean level differences. But I was really trying to get at here was more the like more vulnerable regions that should show clear distinctions, and seeing that that's only happening in Latinx and white participants compared to Black.
Yeah, the way it is it's probably like estimation of the effect.
Definitely. Exactly. Yeah. Right, exactly. Because even like for the white matter hyperintensities, so this was whole brain white matter hyperintensities. And when I looked at this for like parietal white matter tends to be more detrimental, that we see similar, like a stronger relationship there versus like whole brain. But this was just more robust in some ways because just more power. Yeah.
I think a lot of us in here have a lot of intuitions about like the limitations of survey data, some of the features of research design, administrative data. But capturing molecules in large magnetic fields and shaking them a little bit is something that maybe we don't have clear intuitions about. I wonder, as a person trying to study these processes, that if you could, you would be looking at cells collectively, and instead of looking at voxels and stuff. I'm wondering what you see as some of the challenges and areas going forward that you hope that physicists and other people will help evolve.
That's a really good question. I think similar to like when I say as a neuroscientist, it's hard to think about why I'm trying not to use neuroimaging as my sole method of analysis, because I do understand that there's a lot of limitation, not only just access, but also, so what if the amygdala is more active for one group than the other? Like, I still want to understand the mechanism, and I need more than just a brain region performance, whether structure or function, to understand that. So, but I think all these methods have all their limitations. So whether I start doing proteomics and I try to add in more blood data, like, but I think my goal would be that we not focus on which is the perfect method, but more so how can we combine several methods to get at different aspects of the problem and not necessarily pick the ones that suit you better, but maybe which ones can give me a different answer, because we're trying to study the whole body. And so a lot of times we're focused on just the brain, but like everything affects the brain, and it's a feedback loop in some ways. And even though there's some things that don't cross the blood-brain barrier, it affects other parts of the body that does affect cross the blood-brain barrier. So I think if anything, it's more so focusing on like, how can I combine multiple methods and take the best parts of each that give me a different insight into the problem. Does that make sense? Yeah, thank you.
Okay, I may be, you may have covered this, so, but I'm a little,
Oh, no, please.
I'm wondering about selective mortality. There's certain groups, because death rates are also different. And my understanding of EPO4, which could be completely wrong, is that it has something also to do with regulation of cardiovascular and so on. So I was just wondering if, like, how that plays in. And you may have, I things about it, but I was
No, I think that's a really good point. So with the ApoE4, so when I showed ApoE4 here, this is kind of just highlighting like, yes, even though Black participants have a higher rate of having the allele, it doesn't necessarily predict their risk for dementia, even though, because a lot of times you think more of this is bad, so it should predict the worst outcome, but it only does that for certain groups of people. And so to your point, ApoE4 is very much based on different sites of how they interact with other genes, but it's very highly influenced by the environment. And so it's a lipid protein, right? And so knowing that Black individuals tend to have a higher risk of cardiovascular disease, I think the interactions of ApoE4 and their life experiences, it just tends to manifest differently, express differently, but I don't think ApoE4 is the problem. It's like, what's the problem that caused it to malfunction? So ApoE4 won't predict the worst thing, but what's the other downstream, or maybe before any kind of like disruption that's causing it to be that way. So that's why it may not be as predictive in these individuals. And as far as mortality, I'm not sure how, I think this has been shown across even younger adults versus older adults, that ApoE4, you may have a higher rate of it, but it doesn't predict your health as much in certain groups. However, when I showed the allostatic load here, when I say that this disparity was widening as people got older, part of this is because the people who are like, these Black women are not comparable to the Black women who are in their 40s, who would have probably already died off. And so the people who are living, and this is only 65, but we've seen this even much more, a wider disparity in like 85, 90, because the people who are living, because, you know, like there is a difference in mortality rate across race. And so when we're looking at these older groups, it's not representative of the actual life experiences of the average person within that group for Black and Latinx participants, which is more reason I'm really kind of gearing towards doing earlier life analysis, because there's just so many other things we have to account for once we get past 65. Yeah, but there's still very valuable data in individuals who are 65 and older. It's just that it's more reasons to not look at across race differences, but within race differences as individuals get older. Because then the other 85-year-olds would compare to each other, but not across race, because they're just not comparable.
Great,
Yeah.
you so much.
Yeah, thank you.
I actually wanted to ask a question about the ApoE4 findings that you had on connectivity. And does it mean that there is potentially higher reserve among the Black participants?
So I thought that, and so, because there's some literature suggest that, and I didn't have the data to look at like ABC, A7, like more predictive genetic, to see if that maybe would have shown a deficit. But so even like looking at ApoE2, there was no differences across race, like who had more supposedly the protective allele. So I didn't feel comfortable making that conclusion. But I do think, again, like this is why like I want to look at some of the risk factors, but also protective, because there's obviously something going on here that interacts with ApoE4 carriers that make them have better connectivity. However, in this case, better connectivity doesn't always mean better brain health. It should, in theory, but if when I try to relate these findings to cognition, they didn't pan out. So like if I try to look at better connectivity in the ApoE negative, are they doing better cognitively than the ApoE negative white participants? And there was no difference. And so it gets tricky. But again, this is the issue with like trying to use some biological data to like predict like cognitive data and assuming like they go hand in hand always, but there's just so many other variables that we need to account for. So this was just kind of a stab just to show like it's more than genetics for Black and Latinx participants. And as you can see, like the Latinx participants and almost everything I showed today are kind of always just in the middle. And that's partly because they're like very admixed. Like there's so many other factors. Like you have white Hispanics, Black Hispanics, you have different colors, skin color. Like it's just so many things that go into that. And again, it just goes into like different life experiences for me. Yeah.
I think even that was actually one of my questions, thinking about your sample was from like Upper Manhattan, right? So it's like Harlem, right? And so I guess I was wondering, how do you think about, I think there's a lot of sociologists in the room, right? How do we think about like the specific context of that area with how we understand your findings?
Yeah, no, that's a really excellent question. So there's other individuals in the lab who focuses more on the Latinx group, because honestly, they're a whole study by themselves. But like, so some of the things I've kind of taken a stab at, not here, but like for example, the SES findings is looking at English speaking Hispanic participants versus non-English speaking. Most of these Hispanic participants are from the Dominican Republic. So looking at the few that are from Cuba, again, it's just different life experiences, trying to understand like what's driving some of these things, right? So I think language is kind of predictive of like differential experiences. You also see like SES differences by English speaking Spanish speakers versus non-English speaking Spanish speakers. So that's the most I've kind of delved into it a lot, especially because a lot of like these Latinx participants in YCAP, on average, education is 7 years. So that in itself tells us like, you know, it's representative of the participants, but what does it mean to have 7 years of experience in an English-speaking country, and you're showing, in this case, in some cases, better brain health than some of these individuals? And so it's like, why? And so it's just really trying to get at more of those contextual clues. It's just not something I've delved into. I'm more delving into like the Black participants, but that's definitely something that we've been exploring in the lab as well, and seeing some preliminary stuff showing so many other factors that contribute, because we always are like, oh, Latinx, and normally here the Latinx participants does not include Black or so the Black participants are non-Hispanic. So we remove the Hispanic participants from the white and Black group, but this Latinx group could have so many other races in there. Different, like I said, different skin color. So looking at how different skin color affects your life experience as a Hispanic person. Because I mean, it's just a label, but like, unlike Black and White, it's a little more, it's way more admixed with just like the outward physical appearance of their race, that it's just really hard to like put them in one group. So that's why I don't really talk about it a whole lot, but yeah, that's a really great point.
So this is a question I'm just curious about, just from an interdisciplinary perspective, right? Because a lot of us in this room are sociologists, some of us do interdisciplinary work. And from your perspective, because you're coming from a much more kind of clinical, psychiatric context, and you're talking about things that for us are very sociological in terms of what are some of the determinants of things that interact. And what is your experience like being somebody who clearly has like a sociological imagination, and how you're thinking about this and why you would be seeing these kinds of things are. Can you talk a little bit about what that's like? Are you like non-normative?
No, I think like that's a really good observation, in that I think, so even going back to like my story, like when I got into neuroscience, it wasn't because I loved the brain. It was because my grandmother had dementia. And for me, it was like, I'm her favorite granddaughter. She didn't remember who I am. And I'm like, what is causing this? Like, this woman was this like amazing woman. All of a sudden, she's like this very different person. And so I'm like, it's not just biology. And at the time, I mean, I'm like 11 years old. I didn't know what's going on, but we didn't know it's dementia. Back then, everyone referred to it as like senility. Old people just get crazy when they get older. And so I'm like, but what is causing, not everyone's grandmother got dementia. And so I'm like, we're in the same community, but I also grew up like very poor. So like, I'm like, is it the resources? And then also seeing like how, instead of going to like healthcare professionals, we went to like a obia man, like a witch doctor. And so like, although I do think there's some value in like cultural specific methods of getting answers, but it was the distrust in the medical system and seeing how that made things deteriorate even faster. And so then I started thinking like, okay, but what are the structural things in place? Like we don't trust the healthcare system or science for a reason, but it's like there's a structure in place that created that thinking, that experience. And so I'm thinking of like other people who don't have dementia, or the Caribbean is also one of the places that have many centenarians. So like people live past 100, but here she's at 65 with dementia, doesn't make it past 75. And I'm just like, this is not just a biological issue, there's life experiences. Because seeing how, the deterioration happened also very quickly once she stopped going to the garden, once she stopped like just living. It's funny because we had this conversation earlier, but just like living on the farm and just being a really wonderful woman who just living her best life. But when she like fell and couldn't like move around a lot anymore, and then seeing how, of course at the time I'm not thinking this, but looking back, like just thinking of like how all these like life course things that protected her because of probably her upbringing and seeing when it started deteriorating, and also looking at other people in the community and seeing when they started showing symptoms of dementia and trying to understand why was it so common for some people and others. And I was like, it has to be something that's happening with the life experience. And also understanding coming to like America, then seeing like how knowing that this is more prevalent in Black people. And I'm like, okay, but what is in place that, like, what is it? There's nothing biologically reason for like, okay, I'm a neuroscientist, but there's nothing I see in the brain that says a Black person's brain is different from a white person. So why are you at higher risk? It has to be your life experience. So I think like that's kind of where my thinking is. And it's been hard. It's hard to be in a room full of, I always love my talks in a room full of interdisciplinary folks versus neuroscientists, because the questions that I guess is like, well, let's understand something about the brain and this mechanism. And I'm like, I get that, but like, I need to understand the bigger picture in the context, because context really matters. Because at the end of the biological, we're all the same, but we have very different contexts, different experiences. So it's hard, but I love it.